Now, David will be giving this keynote, and he's currently director of the Max Planck Institute for Empirical Aesthetics in Frankfurt since last year. And the big question is, how did he get there? He started in college with math and philosophy, then converged that to cognitive science, cognitive neuroscience, did his PhD at MIT, did a brief stint at UCSF, and then moved to Maryland for 10 very productive years before he was appointed professor at NYU. And he's still continuing running that lab. His main interest is the neuroscience of auditory cognition. That's mainly speech perception. That is also why he's very much interested in time-resolved tools, most importantly MEG, but he's also using other techniques to narrow in on those topics and to get not only a localistic but a mechanistic understanding. And it's not only speech, it also extends to music. It also extends, check out a paper from last year, to screams. Now, he's publishing in excellent journals, but one of his most remarkable papers is actually from the first year of his productivity, which started 20 years ago, and appeared in Brain and Language. And it's quite telling that even last year he was still into screams, because he really started out as a punk. I mean, these days he's clearly establishment, but that paper at the time was very interesting because there were fairly few studies on the neuroanatomy of speech perception, and David tackled them head on. And I perceived that a bit from the other side. So this, I think, is also instrumental in showing us how research can evolve. It was a critique of methods, it was a critique of concepts, of theory, and by all that followed, he really made science advance, and this is really what he has continued doing ever since. So how do you choose a keynote speaker? Obviously, these issues count a lot, but there are two things about David which I would like to highlight and which make him a perfect keynote speaker. He's inspired and he's funny. So please join me in welcoming him to stage to give his keynote.
Thank you so much, Andreas, for your nice, kind, and you were very kind. I know you could have been harsher with me, and I know I often deserve it. Thank you so much to the organizers for inviting me. It's an honor, it's a pleasure. I'm surprised you're here on a beautiful day. What a week. Unbelievable, right? Brexit, Trump still a candidate. What's up with that? My wife's birthday. You know, just really tough. I mean, emotionally very intense. I can't believe we've actually earned a drink. So this meeting is, of course, a remarkable testament to the progress that's been made technically, conceptually on brain imaging. But I think it's worth remembering that we have a historical context. If this button press works, it does. Note where we came from. A long time ago, the idea of sticking people in machines, maybe curatively, to see what comes out was already used. And what makes this picture particularly disturbing is how similar it looks to current labs. For instance, take this one from my lab. You should be deeply afraid. Now, the notion of mapping, in the broadest sense, of course, in mapping language or cognition more broadly, has this fascinating tradition. And one of the first scanners was, of course, Franz Josef Gall. Now, the equipment he used was relatively simple. All of you have this equipment, more or less. He used the hand scanner. Now, hand scanning led to some interesting results, and of course, all of you know it from reading or studying. It led to some things that are actually not particularly helpful. So he had the idea of phrenological mapping, that is, bumps on the head correlating with psychology, and that is what you would call a bad idea. But you should read the book to the end, because he also did some other stuff. Namely, he developed something called the organology, and that is actually a good idea. Now, what is that? It's the presupposition that the mind and the brain actually have a parts list. And in fact, two of the ideas that are due to Gall are at the very core of basically everything we do in psychology and neuroscience. So, two enduring ideas that you should not forget are faculty psychology, or more informally speaking, the mind has a parts list. That's not a trivial conclusion. I mean, we take that for granted now, but the fact of the matter is that was a hard-fought conceptual victory. The second idea of, many careers are built on, and actually the last session showed a lot of that, is the notion of experience-dependent plasticity. That is the notion that using the parts changes their neuronal realization, another extremely interesting concept. Now, this concept of making maps, of course, is very powerful, and it's one of the three kind of larger topics that I want to talk about this afternoon. So the idea is I want to talk about the maps problem, that is, what are we actually localizing? The mapping problem, just to be confusing, is what are appropriate linking hypotheses between different domains, as domains of psychology or cognitive science and neuroscience. And finally, the experimental granularity problem, that is, how are we doing experiments these days, and are they going to inform our theories? So let's get right into the problems associated with the maps issue. Now, for 25 years or more, the principal issue that we've pursued is making maps, and borrowing an expression from the philosopher Kant, you might call it the cartographic imperative instead of the categorical imperative. The cartographic imperative is to try to make spatial maps. It's been very productive and interesting, and it's been at the core of what we do. Now, when we study language, we, of course, are a little bit more clever than it was in the Gall time. We know that language is not monolithic. It has a pretty detailed parts list. So if you've ever taken an introductory course, you know that there are things like the sound structure, word structure, syntactic structure, the prosody, that is the intonation contour, discoursing, so many different parts. So that's good. So one intuition you might pursue is, well, since neuroscience has told us that there are approaches like retinotopic mapping or tonotopic mapping or somatotopic mapping, maybe there is language-otopic mapping. So maybe you just take these domains of experience and you plop them onto a brain. That's not a crazy hypothesis. We even have a journal for that, human brain map. And so we might take domains of linguistic experience and plop them onto brains. That's sort of what we do, and it's not a terrible idea. The question is that the right level of analysis that you really want? So how naive are we willing to pursue this research program? So take a slightly different approach. Take the approach of vision. I should say, by the way, my most vicious critics are sitting in the front, and my friends and critics, and they're playing Talk Tinder. So when I get boring, they swipe. So I keep going. So thank you, gentlemen. I've never heard of Talk Tinder, but I think it's a good idea. So in that spirit, go. How are we to approach these kinds of problems? So take the visual system. Take the classic picture of that we all see in many lectures of the architecture and connectivity of the visual system, a classic, iconic image in neuroscience. Very complicated, lots of internal structure. How about the auditory system? Many different parts, right? So you can see the map on the right was clearly made by someone who studies the brainstem, because it's got many different parts for the subcortical nuclei. And on the upper left, it's got one little sign that says auditory cortex for everything else, speech, music, and so on. But you can see that there's lots of internal structure, lots of anatomical richness. So it should not surprise you that when we go to the language talk, this should not be sufficient anymore. So if there's one lesson, one thing to remember for those of you who aren't in this particular business, it's that this is a very influential, important model. That is, we have the famous Broca's area, the famous Wernicke's area, we have a fiber bundle connecting them, and that's all very nice and good, but it's time to put that to rest. That's been good, and we need to move on now. So how do we move on? Well, a very useful way is to look at the very influential work by Katrin Amons, Zillis, Angela Friedrizi, and colleagues on actually fractionating anatomically what was traditionally called Broca's area. I mean, if you look at the nitty-gritty, it's actually extremely internally complicated and structured. It's got maybe 10, maybe 12, maybe more areas, not even including any laminar issues that might be differently processed. So do you really want to make a functional generalization about one big chunk of tissue like that? Well, clearly not. You wouldn't do it for the visual system, so why would you do it for the language system? Right? So in the visual system, you're willing to say that there are 35 to 50 different territories. So, it's hardly plausible that it should be a much simpler system. So as a consequence, monolithic generalizations about function are not even wrong. Just don't make them. What about the more posterior aspects of the system? So take a look at, in this case, the superior temporal sulcus. A nice, very useful review by Einat Liebenthal shows you just the numerous different functions associated with chunks of the superior temporal sulcus. There's a lot going on. There's a lot of territory used for many different functions. So, okay, that's got to be complicated. So it's complicated in front. It's complicated in back. What about the wire? Well, the wire is also pretty complicated. So here's a let's say, party-neutral review by Dick, Bernal, and Tremblay on different streams. In this particular case, reviewing the several different dorsal streams projecting connectivity. This, I guess, speaks to a debate that I heard was yesterday had between Josef Rauschecker and Angela Friedrizi in another session. Clearly, there's controversy about this, but I think data like these settle the issue. Now, what are we learning? What are we taking away from maps? Well, there are many maps, right? So the maps, very complicated, a rich anatomic parts list. And I myself have contributed to this by adding, with my colleague Greg Hickok, models of how speech processing is organized or how meaning is organized. That's all very nice and good. We have networks of areas. And what we should remember from this is that the maps problem, that is making detailed functional anatomic maps, is an important part of the homework, but it's really only part of the homework. It's not actually the answer. And the message, the one point I want to sort of leave didactically is localization is not explanation. We all know this, and yet we all pretend it's true. I'm about to show you one of my own experiments. It makes me eat my own words, right? So localization is not explanation. It's part of the way. It's just part of the homework. So let's go into this from a particular point of example. So recent paper, very interesting, by Hoot and colleagues, a tour de force of engineering, and a very interesting set of results. The experiment, pretty straightforward. 7 people listened to a couple of hours of stories. And then the analysis, a very sophisticated analytic approach, simply used kind of statistical co-occurrence of words and word frequencies in probing brain activation. And what you see is the kind of plot below, which is, it's a little bit difficult to interpret, but I'll show you the cartoon summary of this. So from this kind of analysis, they say, well, the co-occurrence used leads to semantic features, a laudable goal, very interesting analysis. The result leads to this, what they call the brain dictionary. And you can see all over the place, in both hemispheres, in fact, words distributed. Now, that's a very interesting result. I mean, and it's the finding based on really sophisticated, cutting-edge engineering. But based on this, how would you change, or how would you inform your theory of meaning, semantics, cognitive psychology, or anything? So that's, for me, the challenge here. That is, it's very hard to see how this very sophisticated finding connects to anything we know about language processing. So let me give you a brief example, something that you're actually good at. So, here are individual words everywhere, and this is, a comprehensive atlas of meaning. Here's one word that you can all recognize immediately, unflushable. So suppose I say the sentence, my iPhone is unflushable. You know immediately what this means. You don't have to work very hard. You extract the correct items, put them together, and interpret the sentence. Like, don't put this in the toilet. Right? This is good. Please don't. Now, how can you do that? Well, you need a pretty detailed processing model, right? So you need to know, how do you get these individual parts? You can't. So if I say this iPhone is unflushable, there's no statistical information to draw on. It's a very rare sentence. The transition probability is essentially 0. So you can't draw on that. So you must draw on an analysis of the local items and their structure. So you have to look at flush. Maybe flush is somewhere here, hopefully. Then you have to look at able. Well, where is able? And then you have to look at un, which is called, by the way, unprefixation. Very useful. Like this lecture is unbearable, for instance. Now, that is not to say that that's not possible, but it's to say even a very sophisticated analytic approach, like the one in this recent work, is actually not able to account for elementary aspects of language processing unless it's connected in a principled way to some computational model of linguistics. It's just not possible. So even an example like unflushable needs to be tackled. These are the things our mind is good at. Now, having said that, let me now eat my own words and give you an example of making exactly the same mistakes. So let me give you a quick tour of an experiment that addresses a mapping issue. Namely, here's the model of speech recognition that Greg Hickok and I have worked on, and we're interested here in one specific question. There's two, the red box surrounds two functional boxes, one that has to do with a spectrotemporal analysis of sound, and one that's allegedly about speech sounds in particular, phonology. So you might ask yourself, why are there two boxes here? Why is this guy having two boxes? Maybe this should only be one box because that's the much easier theory. So, on any model, is there a yellow box? So the experiment I'll show you very quickly asks the question, is there a yellow box? And it's a work done by Tobias Overath at Duke, Josh McDermott at MIT, and Jean Zarate at Nature Publishing Group. And here's the design of this experiment. We borrowed or adopted and adapted a design of an older paper by Kalanit Gross-Spector in Vision. And the idea of that experiment was the following. If you look in the top row, on the upper left, there's a picture of a cat, because the internet has a lot of cat pictures. We know this. Now, if you take the cat picture and you chop it up ever finer, you get kind of higher spatial frequencies, but less and less aspect of objectness. You can't see any objects anymore. Now, when you present that to participants in an fMRI experiment, you can see, depending on where you probe in the visual hierarchy, a degree of selectivity. That is, if you look at V1, all these different images, 1 through 5, give a pretty good response. But if you look at, let's say, the lateral occipital complex, you see it gets increasing selectivity for bigger chunks that are like an object. You get the general idea? So if you see parts of an object, that part of the brain is happy, right? This is hardly surprising. Now, how can you do this in an auditory version? So we made an auditory stimulus which we call quilts. Quilts are these lovely blankets which you stitch together. And let's see if so actually, how do I maneuver to the sound? Can someone help me with that? How can I play the sound? There must be. The tech support has to click for me. Okay, tech support, can you click the upper left? Thank you. Please. This should confuse you. Good, you're healthy. So this was a long sentence of actually German from a bunch of different speakers, chopped up into short segments, 30 millisecond segments, and then completely shuffled, and played to listeners of American English who are not very well educated, NYU undergraduates. So they didn't really understand anything. Now, tech support, can you play the upper right sound, please? Upper right.
Als Homo-Duplex-Koordinatensystem X eingezeichnet. Übrigens klassifikatorisch aufgeklärtes Standard dagegen suchen.
So, those of you who speak some German recognize little chunks of words, presumably, right? So that's because these are much longer segments. So we did this for a range of things. The victims were in the scanner. We presented them these annoying sounds and probed those parts of the auditory system known to be sensitive to different temporal structure of speech and so on. So we look at for instance, the Heschel's gyrus, so the home of core auditory areas, the planum, the superior temporal sulcus. We define these both anatomically and functionally with the participants, and then play these sounds. And the hypothesis is the following. If there are cells that are tuned to speech signals, that is, if there's a yellow box, that activation should increase as a function of the segment size. Because if the segment size gets bigger, you have more speechy information as the statistics of natural speech are increased. So is that true? The answer is absolutely. Damn right it's true. So take a look at, here's the imaging findings from the left and the right. So the red line, again, is the Heschel's gyrus. So that shows you that primary auditory areas like all kinds of sounds that have any kind of modulation, right? So there's no differential sensitivity as a function of the rate, which is at the bottom. The blue line, very similar. not much change as a function of segment duration. But if you look at the green and black lines, which are the functional regions, very high sensitivity to this manipulation. So what's the message? The longer the segment, that is, the more speechy information you have, the larger the activation in both sides. Okay, so that shows us something interesting, namely that there's a high sensitivity for that kind of information there. Okay, cool. Now you should ask, does it have the right specificity? So we now went on a orgy of controls. I mean, a perverse experiment. I'll just give you very quickly just a couple of, maybe two highlights. So when you listen to these sounds, they sounded weird, and not even good kind of weird, like techno Berlin weird, just annoying. So that's because we changed them, we manipulated them in a way to change temporal structure. So now we did a few others, which I'll play you now. Hang on here. So suppose you make a sound that's not a speech sound, that's a textured stimulus, but it's actually optimized channel by channel in the auditory system to have the same modulation property. So it's exactly like a speech sound from the physical point of view of the modulation across channels. And now if you listen to this, it gets a little creepy. So tech support, if you could play the third, that one, yes, good. So that's just annoying again. Now tech support, the one on the right. So now if I put a lot of pressure on you and I ask you, is that speech or non-speech? A lot of you would say, well, that's like a crappy cell phone signal. That's sort of speech-like. It's pretty close. But of course, it's nothing. It's just a texture, right? It's made from basically a form of noise. But it sounds pretty close, so it should make you a little nervous. So now you're reviewer number 3, and you say, okay, that's a nice control for kind of the amplitude modulation properties of sound. I can live with that. But really what's going on is the sensitivity isn't to these weird parts of speech. It's because you have complicated sounds that have meaning. You say, well, that's maybe true. Let me tell you, of course, that the pattern shows on the lower right, the imaging pattern shows you first replicate that the subjects get the speech pattern. Auditory cortex likes everything. STS likes only the speechy stuff. And the right side are these co-modulations, so not interested. So now let's respond to reviewer, was it 2, 3? I don't remember. So let's say I want to see you do an environmental sound. We can say, okay, fair point. Good objection. Play the upper left. Okay, and now all the way over. Yes, that one. You get that? Okay, that's not tennis. It's untennis. Now, how about the one in the middle on the left? The middle on the left. Yeah. All right, and the middle on the right, please. Okay, you get the general idea. So the same temporal manipulation or quilting manipulation is done now on a wide variety of environmental signals. A new batch of victims is brought to the lab. They undergo the same experiment, and again, same thing happens. So you see on the left, Heschel's gyrus likes everything. It likes, you replicate that it likes the speech things, and you show that it likes environmental sounds of all forms, but the STS actually does not care. So it's not sensitive to modulation. It's not sensitive to co-modulation. It's not sensitive to environmental sounds. Let's do one more for good measure because it's such fun. What else could it be? When you have longer segments of sound, what else changes? Well, the amount of intonation contour, right? So the amount of prosodic modulation per unit time changes. So let's do how can we test this? We can make another stimulus in which we retain the speech and get rid of the pitch information. It's called a noise-vocoded speech. I'm obviously really violent with this thing. I got to bind my hand back. So let's see what that sounds like. Okay, tech support, lowest on the left. Lowest, 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 lower, lower, yes. That's what it sounds like when the United person says there's a huge delay and they're not telling you why. That's sort of like classic. Okay, now the lowest on the right. Listen, German speaker.
Vielfalt der menschlichen aufgeklärte Standard- und Seelenkunde in sich Verhaltensweisen verfielen zu Sprachen als Homo dupla.
Okay, so German speakers can actually extract stuff and understand that just fine. Okay, so now we don't have pitch information, but we do have speech information. So now the casus knaxus. What does it go with? Does it show sensitivity to pitch or sensitivity to speech? And the answer is speech, of course. So now you look at the right side, the STS actually shows as you give more longer segments, you get the extremely heightened sensitivity to speech. This is wonderful. So we can conclude from this, and it's all along the STS, no localized specialization. You can read all the minutiae and the endless additional controls in a lovely paper. Highly recommended. 5 stars on Goodreads. So what do we conclude from this? Well, this is another example of a mapping study, right? So we used quilts, which are a very useful acoustic stimulus. We observed from an imaging point of view, from a human brain mapping point of view, that the STS bilaterally has a very high sensitivity to speech. It has a high specificity to speech. And in a bunch of additional experiments, which I won't tell you about, it has an intrinsic time constant that's below half a second. Now, what do we conclude from that? Well, we started by trying to test a hypothesis. The hypothesis was, is there a yellow box? That is, do we make a differentiation between a spectrotemporal analysis, that is an acoustic analysis of sound, and one that actually builds speech units, elementary units of perceptual analysis? And the answer is yes, there is a yellow box. Okay, so that's a useful thing. But of course, I still have to eat my own words, because still, it's not an explanation. It's a localization of a series of processes, but it doesn't tell me in any mechanistically satisfying way what's going on. It tells me a location and some broad hints. So the mapping is useful as an intermediate homework step, but we must go further. That is the message of this part. Now let me try to tackle it in a slightly different way. Let me switch gears to what I call the mapping problem, just to confuse myself and you. Because mapping is now no longer making a map, a spatial map, but it's making a mapping from one domain to another. So what's the intuition behind this? Well, this is what we all do for a living. We're basically like car mechanics. In this case, Volkswagen, for good reasons, needs to be taken apart. And this is all the parts that make up a Volkswagen Golf, right? So this is the entire parts list of a car. And this is what we essentially do, whether we're neuroscientists or cognitive scientists or linguists. We try to figure out what are the elementary particles, what are the atoms of neuroscience or the atoms of language. Now, take the right side column here. If I go to the Society for Neuroscience and I ask everyone, what is the elementary parts list for you? You say, well, dendrite or neuron or LTP is an elementary operation and so on. There's going to be some, disagreement, but some agreement. That's the parts list of neurobiology. Now, if I do the same thing at the Linguistic Society of America or the Deutsche Gesellschaft für Sprachforschung or God knows where, you get a very different parts list. You get the parts list of language sciences, like, for instance, the notion of a distinctive feature or a morpheme or the notion of concatenation and so on. Now, that's all nice and good, and for both of these sides, for column A and column B, there is a ton and ton of evidence. This isn't just, made up overnight over beers. I mean, all of these things are, supported by dozens and dozens of studies. So our job, ostensibly, is if they both have explanatory value, is how do we link them? And the fact of the matter is we don't even have the slightest idea. I mean, not the slightest, because we don't know how information is represented, right? So it's unclear how to link these domains or how to map them onto each other. And the reason is, presumably, because we don't have the kind of linking hypotheses by which we could actually explore how brain mechanisms form the basis for this kind of computation. That's our aspirational goal. That is, we want to align the alphabets, the alphabet of domain A and the alphabet of domain B. But we don't know what the right way to ask the question is. That's, at the moment, it's largely correlational. We do something here, and something here happens, and we say, oh, it correlates pretty well after correction. Okay. It's not so satisfying. It's probably not good enough. Now, you might say, oh, you should be more reductionist, because we know everything about neurobiology. So take C. elegans. That's a beast where we really know everything. We know the neurons, we know the connections, we know the genome. It's a beautiful case where you should be able to understand the entire beast because we know the wiring diagram. But this is a terribly sad and annoying object lesson for all of us, because in C. elegans, the fact of the matter is we don't know anything about why that creature moves and does anything it does. We have the full understanding of this thing and don't understand it correctly. So I'd like to read a pretty useful quote from my colleague Movshan. I think it's fair to say that our understanding of the worm has not been materially enhanced by having that connectome available to us. We don't have a comprehensive model of how the worm's nervous system actually produces the behaviors. What we have is a sort of bed on which we can build experiments, and many people have built many elegant experiments on that bed, but that connectome by itself has not explained anything. Now, that should worry you. I mean, it certainly worries me, because it suggests that the deep knowledge of the wiring diagram is insufficient. We need more. So what do? Well, we try to tackle the question of linking hypotheses. So one way to go would be, as many of us in our area of research do, is to try to decompose or fractionate what we know about neuroscience and what we know about language processing into kind of more element computational primitives that actually might be part of such a wiring diagram. So take the stuff on the left, like all the parts of linguistics, or the parts on the right, the parts of neuroscience. Maybe we can break those down further. We can decompose them into the kind of elements about which we could formulate a linking hypothesis that's not crazy, or maybe crazy but not insane, just hopefully productively wrong. So one might be, so if you study language, you might say, well, one thing you better be able to do is, for instance, concatenate elements or build constituents, very elementary things. So that's the kind of elementary computation. If you don't have a story for, you're in really lousy shape. So let me give you one other experiment that tries to tackle this particular question, very, very briefly. So this is an experiment run by primarily Nai Ding and Lucia Meloni, two quite extraordinary young scientists. And the experiment builds on the following observation. So when you listen to a speech signal, like me blathering on, Tindering myself, everyone has an intuition about the bottom, right? The bottom is a spectrogram. The x-axis is time, the y-axis is frequency, and what we all learned when we took a psychology course or linguistics or acoustic phonetics is that you look and there's spectral information. There's burst information and form and transitions and vocalic patterns and so on. But if you look at the top, there's something called the speech envelopes, basically the amount of energy per unit time as it changes. And we have not paid so much attention to that. So it turns out in the last 5 to 10 years, a lot of research has been focused on the speech envelope and its potential contributions. And through a bunch of experiments in many different labs, one really fundamental observation is now clear. That is, the auditory cortex actually entrains to the envelope. And for reasons that I don't have time to explain to you, what that means is that basically you entrain to the syllabic rhythm of speech. That is, the envelope, the brain waves surf on the speech waves in kind of a literal sense. So there's speech waves like this, and the intrinsic brain activity surfs on that. And you might ask, what's that good for? Does that do anything? And a nice way to think about it is a kind of physiological model. So Annelise Giraud and I published a more detailed physiological exploration of what these oscillations do in parsing speech. And to make it a little bit more kind of intuitively pleasing, here's a way to think about it. What this brain activity does is it creates elementary events. So it parses events. So if you think about a typical spoken sentence like, she had your dark suit and greasy wash water all year. And it's one of those classic sentences from a battery that we use. And notice when you hear it, just like it's written there are no white spaces. Unlike the sentence above, parsing events. There's nice white spaces, so reading is much easier than hearing. Now what happens, here's the sentence. What happens is the brain waves, these low-frequency brain waves, attach or surf on the energy fluctuations. And what that means functionally is it breaks it apart and adds little white spaces. So it parses or segments the signal for then giving the right chunks for decoding. So it gives you kind of a two-stage possibility. Now, there's lots of interesting evidence for this. Now, you might ask yourself, well, that's at the periphery, and that's an interesting mechanism. So use intrinsic neural oscillations to parse the system into elementary parts. Can you use this same system for something a little more interesting? Because I'm not interested in just hearing syllables, ba, blah, blah. I want to actually put words together and extract meaning. That's a more laudable goal. So the question is, can you use a similar mechanisms? Now, the boundaries between syllables actually are pretty well defined between these amplitude envelopes. It's a good heuristic. But of course, in sentences or words, there is no acoustic information of that form. That is, there's no, because we don't speak like this. It would be too irritating. Now, so it can't be some obvious physical signal. But if we know that there's a neural code for linguistic levels at the periphery, that is, you entrain to this information, the question is, what is the neural code at the higher levels, and does it change at the same rate? So if we know there's entrainment to the periphery, that is, we entrain to the envelope and therefore the syllabic structure, do you also entrain to the hierarchical linguistic structure that's there? That is, do you recycle the same computational mechanism to do structure building operations? That is the question. So now I'll give you a very quick tour of a quite simple experiment, but I think it's quite fun and has good audio examples. So we built a little study, doing it first in Chinese and then in English, and the study has the following properties. All the sentences are presented in isochrony, so at a completely fixed rhythm. So for instance, dry fur, rubbed skin, new plans gave hope, and so on. But they're built so that if you move them around, they're either just a word list, or you shuffle them so they create little structures, that is phrase structures or even sentential structures. So let me play you an example of this for a second. Can you play the upper sound?
朋友请客，汽车拐弯，青草发芽，飞机降落，游客爬山，母鸡下蛋，灯泡发光，英雄救火，骏马奔驰，农民种菜。
Right. That's what she said. Now, if you're a speaker of Mandarin, you presumably recognize some sentences. Now, the question is, what do you entrain to? Because if we shuffled it the right way, of course you should entrain to the syllabic rhythm, because that's the stimulus. But what about the other information? And so we did this experiment first with Chinese listeners, and here's the answer. The answer is, remarkably, that you actually track all aspects of the structure concurrently, and that's done when you analyze the frequency domain. So if you look at the peak, so the thing on the left, the little cherries, each of the round cherries is one of the syllables, which is also a word in Mandarin. And then 2 together are a phrase, and the 4 are a sentence. So, but this can be shuffled. So now you see that the syllabic rate actually gives a big peak at the rate the syllables were presented at, but you also see peaks at the phrasal rate and the sentential rate. But you have to be impressed by that, because those are not in the signal. Those are there because you had to construct them, right, internally based on your grammatical knowledge. So you might say, oh, I need to know a lot more before I believe you. First of all, when you see this, you should say, wow. This is a wow kind of result, because that does not come for free, because building structure internally based on your grammatical information is not a triviality. I mean, that's a very interesting feat of cognition. So let's go quickly and show a couple of controls. This experiment, too, was an orgy of controls. So first of all, let's just take all those words and mix them up. So same rate, but now they can't build structures because they're not grammatically licensed. Then you see a peak at the syllable rate, but you don't see anything else. Good. So it's not just subharmonics or some physical thing. What about if you just build little pairs, like Angry Birds, Black Table, Long Lecture? Then you get it at both rates, at the rate of the stimulus and the rate of the phrase. Also good. What about if can you get the original finding back? Yes. Can you move the internal parts around? Yes. Okay. Can you make it go away? Play Chinese materials to a American English listener. Yes, they can actually track the auditory waveform. No surprise there, because it's a very robust suprathreshold signal, but they don't get anything associated with the meaning, so it's good. Our English listeners weren't secret Chinese speakers. Now, one more. Yeah, play that. The lower one, please. The lower one.
Poor friends paid bills, our boss wrote notes, drunk dudes sang hums, fun, games, waste time, kind words, warm hearts, tall guys, flea, camp, rude, cats, claw, dogs, quiet, lamb, 8, grass, wood, shelf, holds, cans, iced, beer, cost, cents, new, plans, give, hope, large, ants, built, nests.
So now you get the general idea. It's very weird, and I don't know if you have this experience, but as you start putting them together, you start hallucinating that there's actually a kind of contour. But I assure you there's no contour because each word was recorded and synthesized individually. Right? So there's no acoustic contour there, but you start believing there is one. So does it work in English? Yes, it works in English. Final thing, so why do we care about this? Well, we care because it suggests that cortical activity is entrained to not just the physical signal, that we knew before. That means you just follow a stimulus. But cortical activity, or a similar mechanism, entrains you to the phrasal and sentential rhythms of speech that are not obvious in the physical signal. So you see it cross-linguistically, and you see it despite the fact that there's no acoustic information available for you. So that's pretty neat. Now, one last point might be, well, you say, maybe this is all just statistics. Everything is always statistics, especially at a meeting like this. So we did another bunch of experiments to verify that it's not just transition probability. And let me give you one, hang on. This one, can you play this one?
Dogs can be smart. My cat is so lovely. Amy owns a farm. Homework needs to be done. Nancy works at home. Rumors are not true. He will take the train. Matt married Carol. Earning money is hard. New York never sleeps.
Yes. So now, if you have statistically conditioned information, and if all you were tracking was statistical information rather than structure building operations, then you should simply follow the tracking of statistics. It would be like the red line at the bottom, because you're just tracking probabilistic information that's held constant. What you actually find is that you track the structure. That is, you can't help but build syntactic information, whether you want to or not. It's by rule and not by exception. So we also source localized this, yes. Thank you, reviewer number 4. So what's the message of this? The tracking of linguistic units is grammar-based, and it's not confounded by acoustic information or probabilistic information. That is, you can use these kind of intrinsic oscillatory information as a device to track information concurrently across different hierarchical levels. Now, I think that's pretty cool, and you should think it's pretty cool. And it shows you what's the message. In some sense, the message is you can use, in this case, what were we after? We were trying to show, remember, as part of the mapping part, that you can get elementary computations like structure building or constituent structure alignment through these techniques. And this suggests that it actually works and that maybe we have some candidate mechanism that we can use to exploit this. My last remarks in the last 7 seconds, or maybe 60 seconds or so. What's one of the issues we have? What I call the experimental granularity problem. So a lot of the experiments we do, ultimately aim to do something naturalistic. Right? So this is a typical situation. There's lots of people. It's noisy. It's interesting. It's multisensory. It's contextually dependent. It's interactive. That's what we care about, right? That's at least what we say in our grant proposals. In our grant proposals, we say, oh, the world is multisensory. We need contextual information. It's very important. And of course, it's trivially true. And it's hardly informative to say that. And what we really care about and of course, are connected to emotionally are things like, babies that are sweet, or cute things, or socially interactive things, or awesome things. And now look, I have nothing against awesomeness. I'm for awesomeness. Make awesomeness great again. However, the question is, how do we actually do experiments? And here, there's now a tension. So the tension is that the experiments we recently, we increasingly do in many of our labs are kind of ecologically valid and naturalistic, and I think that's good, and it gets us to an interesting part of psychology. That being said, does it actually work? Now, this is the issue of granularity and mapping, because most of what we know, let's go back to vision, where we know a lot, is not based on relentlessly cute, awesome things. It's based on really boring things like luminance contrast, which one can get excited about, but, or chromatic contrast, like a Mondrian, also very cool. Or Gabor patches, the best, okay? Or moving dots. If you can turn on that video, that's a structure from Motion Demo. If you can click on it, tech support on the lower right. So those are the things, if you just click on the, just so people can enjoy the moving dots. Lower right. There you go. Wow. I mean, it moves me every time. But it's impressive, right? I mean, it's impressive what a visual system can do. But what's the point? The point is that it's experiments on these kind of objects and manipulations that have taught us to learn a lot about the system. Now, that doesn't mean this is the only way to go, but the question is, what balance are we trying to find as a community between fine-grained experimental manipulations of things like luminance, color, motion, spatial frequency, and naturalistic experimentation like the experiment where you're lying in a scanner and listening to a story for 2 hours? I mean, that's very interesting and requires a different kind of analysis. But the models we have at the moment make use of concepts really coming from the cognitive scientists and detailed things. Like, for instance, the concepts of computational visual models are things like view-based module or, Gabor filter or similarity space. Now, those are computational concepts that simply will not come out of simply recording people doing nothing. It's just not conceivable, right? So if those are the atoms of our models, the question is, how do we get to these? And these require granular hypothesis testing-based experimentation. And I have to eat my words here because I have argued that we shouldn't be doing these kind of experiments because it's misleading and it leads us to study only tasks. But now I'm sort of stuck. I feel like we have to, how can we learn by not doing this? So our models of the visual system that are, motivated, computationally explicit, and biologically really well supported, by and large, come from these very detail-oriented manipulative experiments. So, in conclusion, I mean, if you want to answer the big questions, the questions that really link between the neurobiology and cognitive science at large and language processing in particular, regardless of what your predilections are. I'm fanatical about MAR, for instance, on the right. MAR's levels of analysis, I can't help myself. It's sort of like deeply ingrained. I know it's irritating to my friends and family. If you're deeply wed to an ethological perspective a la Tinbergen, or if you're Aristotelian, which is a very reasonable thing to be in a philosophical stance, you will have to answer these questions. That is, the relationship between, let's say, the material cause in Aristotle's sense, or the implementational level of analysis in Mar's sense, and the computation algorithm cannot be left unanswered. And the question is, how will they yield? How will we get the kind of linking hypotheses between brain and cognition that give satisfactory explanatory models and not just descriptions? We don't just want descriptions, we want explanations. And so we're answerable to these very difficult things. So my conclusions are that localization is wonderful, we do it for a living, but we can't mistake it for explanation. We don't actually have linking hypotheses, or very few that are satisfying, and we should actually strive together to have, we should have higher yearning to find them. And context and naturalism are wonderful, but in my view at the moment, they actually make our word harder. So I think we should be radical decompositionalists. So we should make the small, so should we, in that sense, borrow from biology or physics, that is, make the smallest parts. So the point, my resolution, my New Year's resolution, and our overall resolutions are, we have wonderful spatial resolution, and it's getting ever better, and our techniques are, amazing in the last 20 years. It's wonderful. And we have the appropriate temporal resolution, but we still lack conceptual resolution. I think we could do with better conceptual resolution. And I leave you with that. Thank you for coming, thank you for staying, and have a good afternoon. Good.